DNA is made up of nucleotides, which contain a pentose sugar, a phosphate, and a nitrogenous base, all connected by hydrogen bonds. Each base matches up with another base, either adenine, thymine, guanine, or cytosine. These form an anti-parallel double helix structure. Each base matches up with only one other base. This means that parts of the DNA can be easily copied for use in creating proteins. The first step for protein synthesis is transcription, which for eukaryotes takes place in the nucleus. The RNA polymerase latches onto the DNA, starting at a specific base sequence called the promoter, and moves along the template strand of DNA. Each DNA base is matched up with its corresponding base, the only difference being that adenine is paired with uracil instead of thymine. These bases are linked together to form a chain, or messenger RNA. When the RNA polymerase reaches the termination sequence, the mRNA is nearly complete. However, before the mRNA can be used to create proteins, it first has to be spliced. The pre-mRNA has its introns removed, and the remaining exons are spliced together to form mature mRNA that can now exit the nucleus. Though both DNA and RNA contain the instructions to synthesize proteins, DNA contains all the genetic information for organisms, while RNA contains the information needed to build particular protein. DNA is also a double helix, making it more stable than the single-strand RNA. RNA's pentose sugar is ribose, while DNA's is deoxyribose. Also, thymine in DNA is replaced with uracil during transcription.
Once the mRNA leaves the nucleus, translation occurs in the cytoplasm of the cell. First, a ribosome, which is made up of ribosomal RNA, attaches to the mRNA. Translation starts at a specific start codon, which is methionine. The transfer RNA in the cell contains a specific anticodon and carries a type of amino acid. When the type of tRNA is needed based on whether the codon on the mRNA and the anticodon on the tRNA match, the tRNA enters the ribosome and then adds its amino acid to the polypeptide chain. It then leaves to pick up another amino acid. This process continues as the ribosome continues to read the codons and gather the required amino acids. As the mRNA is read and the codons are matched to their correct anticodons, the polypeptide chain is added onto. When the ribosome reaches a stop codon, the polypeptide chain is finished and ready to be formed into a protein. That polypeptide chain, or protein, then goes from being in its primary protein structure to then being a secondary structure, as either a beta-pleated sheet or an alpha helix structure. Once it is a tertiary protein structure, it becomes a mix of both the pleated sheet and/or the alpha helix structure, and in its final form, multiple protein chains in their tertiary form come together to form a quaternary structure. The process of both transcription and translation is the first step to what creates proteins and thus all life as we know it.